Okay, class, I'm going to talk on meiosis. I am not going to do an extensive discussion of this. The only reason I'm bringing this up is to compare it, contrast it to mitosis. Mitosis was in somatic cells. Mitosis was an exact duplication of cells. Meiosis is in gametes. In gametes, you want to take a cell and reduce the genetic material amount so that you have half the genetic amount of material in the sperm and the egg. So when fertilization occurs, they both come together and restore that. So we're only going to hit it from high points, and this is not something to really do a big detail on. It's more in ANP2. So meiosis is nicknamed reduction division. It's a process where the cell divides but reduces the genetic material one half. This type of cell division occurs in gametes. Remember, mitosis occurs in somatic cells. The original parent gamete cell, which would be called a spermatogonium in the male or an orgonium in the female, have 23 pairs of chromosomes, which means diploid. Diploid means twice, which means one set from mother, one from parent. The other, in a mathematical way, is called 2N. So if you say a cell is diploid, that means it has 46 pieces of genetic material, 23 pairs. Or you could say each N is one complete set. One set from mom, one from dad, 2N. If you only have one half set, like you would have in the gametes, is called haploid, or 1N. So the original parent set does have 2N, the goniums. But doing meiosis, you reduce it down to haploid, 1N. Okay? The differences between mitosis and meiosis: mitosis occurs in somatic cells, meiosis occurs in gametes. Mitosis has one nuclear division, meiosis has two. So you actually do two nuclear divisions. Mitosis produces 2 daughter cells. Meiosis produces 4 daughter cells. Each of the daughter cells, though, has one half the genetic material. Okay? So here would be an example of that. Here's mitosis, 2 cells, both 2N. Here's meiosis. You go to a meiosis 1, then a meiosis 2, and then 4 cells, each having half the genetic material amount. So that's a major thing. Now, the other thing I want to bring up is that I'm not going to get into that. I want to bring up this issue here. Let me find it here. That we call crossing over. Okay. You do not look exactly like your parents. You look like a blend of it. 2 reasons for the blend. One is that it depends on how the cell divides as to whether chromosome 1 will come from dad or mom or so forth like that. 2 from dad or mom. But during the process of meiosis, the chromosomes actually break. So on each chromosome, you actually put a piece of dad and mom. You put a piece of dad and mom. In meiosis, what would happen is, if that didn't occur, you'd either have to pick, well, do I want mom's 1, dad's 2, dad's 3, mom's 4, which meant any blueprints on chromosome 1, 2, or 3 would be lost forever. So you would have lost all your inheritance on any one chromosome because you'd had to pick either dad or mom's. In crossing over, which I won't get into, the chromosome actually breaks and recombines and puts a piece of dad on mom and a piece of mom on dad. So each chromosome actually carries a piece of the other parent. That's another issue. That gives evolution, that gives genetic diversity, a whole blending, a whole plethora of things. Okay? As I mentioned, there are 2 nuclear divisions that occur. We won't get into it. Ultimately resulting in 4 cells. each having half the genetic material. Oogenesis makes the egg. Now with the egg, this is what happens. You start out, you do get 4 cells, but 3 of them don't work. Because what you've got to do is put all the food into one cell. Because unlike some animals, you have to have enough food in the egg until the afterbirth develops. You have to have enough food in the egg. See, if you're dealing with a chicken, you hatch the egg, so all the food has to be in the egg forever because there's no connection between the mother and the chicken and the egg. But in a human it is, but the placenta does not develop for a while. So you have to have enough food in the egg to do that. The sperm only brings genetic material. It doesn't bring anything else. So oogenesis, development of the egg. So you'll have 1 egg, 4 cells, but 1 viable egg. The other things are called polar bodies, and they degenerate. That's oogenesis. Okay? That's oogenesis. So you would make 3, you would start the same way. You'd get 4 cells, but 3 degenerate. The ovum gets all the food. Okay, that's the one that you as a female would ovulate and would get fertilized. Okay. In the sperm, though, you actually do make 4 viable sperm because you need a heck of a lot of sperm to fertilize 1 egg. So that's all I'm going to really say on that. So I go back again and I say that the key to this is reduction division to get half the genetic material. It differs from mitosis, as you can see, and you get 2 cells, both with 46 pieces of genetic material. In this, you get 4 cells, each with 1/2. If it's a male, you get 4 sperm eventually. If it's a female, you get 3 polar bodies and 1 egg. Along the way, you do this called crossing over, which rearranges the genetic material so that all the chromosomes will have a piece of mom and dad on them. And I'm going to let that be all I'm going to say on meiosis.